Hey everyone, my name's Tom Laliva. Thanks for tuning in today. I've been studying functional neurology or applied clinical neuroscience and have made some videos for any students that are doing the same. I wanted to provide a resource to break down some of the basic eye movement pathways. So this is going to be one of a few videos I'm doing on the pursuit and saccade pathways. All the information in these videos came from various resources, including textbooks and papers that I've referenced in the next slide, as well as my studies through Dr. Carrick and the Carrick Institute, and learning from my mentors, Dr. Ellis, Dr. Traster, Dr. Longyear, and Dr. Stangl. None of the information is my own original stuff. I've just done my best to make some pretty complex neurology a little bit easier to learn. So I hope you can all gain something and learn something from these videos. This information is always changing as our knowledge and science progresses. So please let me know if you have any thoughts, any feedback, and I hope you enjoy the videos. So in these videos, we're going to be going over some of the basic eye movement pathways. And to introduce the subject, I'm going to read straight from The Neurology of Eye Movements by Lee and Zee. So, during evolution, the first threat to clear vision was posed by head movements, especially those occurring during locomotion. Because our eyes and retinas are attached to our heads, if we had no eye movements, images of the visual world would slip on the retina with every head movement. This would cause our vision to become blurred and our ability to recognize and localize objects to be impaired whenever we move through the environment. To this end, two distinct mechanisms evolved to stabilize images on the retina in general and the fovea in particular during head perturbations. The first compromises the vestibulo-ocular reflexes, which depend on the ability of the labyrinthine mechanoreceptors to sense head accelerations. So this VOR is particularly important when we're walking. Every time we take a step, our heel strikes the ground, we have a shock wave that goes up through our body and moves our head. And if we didn't have the VOR that moves the eyes equal and opposite to the head movement, then the image or the object would become blurry. It's our VOR that keeps those images clear when our head's moving. The second consists of visually mediated reflexes, the optokinetic and smooth pursuit tracking, which depend on the ability of the brain to determine the speed of image drift on the retina. Together, these gaze-stabilizing reflexes keep the fovea of each eye pointed at the object of regard whenever the head is moving. In this first video, we're going to be going over the smooth pursuit pathway. But before that, just a little bit more background. If this is the eye here, and this is the retina, for clear vision of an object, the image must be positioned steadily on this fovea region of the retina. And that's where we have greatest photoreceptor density. And a 2-degree movement off the center of this fovea is associated with a 50% decrease in visual acuity. So ideally, the image is within about 0.5 degrees of the center of this fovea region. And when we're looking at the pursuit pathway, basically a pursuit allows us to maintain the image of a smoothly moving object or target close to this fovea area. And that's the area of highest visual acuity because it has such a great photoreceptor density. So ideally, the pursuit is going to match the motion of the target, but due to the delay of the image or due to the delay in some of the visual processing, we're going to have problems. So what the brain does is that it generates pre-directive smooth pursuits, and these movements can maintain the smooth pursuit as the target kind of drops out of sight or moves out of sight. And not only this, but the objects must be attended to whilst the background or the smeared images of the background must be ignored. And this takes some pretty good brain integrity. We're going to go through how certain areas of the brain accomplish this in the rest of these videos. So let's say we have an object in this left visual field and it's moving on our fovea here. So it gets processed by ganglion cells in the retina, and it relays signals through the optic nerve and the optic tract, passing through the optic chiasm here, and it reaches the lateral geniculate nucleus in the thalamus. We also get projections to the nucleus of the optic tract, which is located in the pretectal area of the midbrain between the two superior colliculi. And this is important, we're going to come back to it. We've got projections from the nucleus of the optic tract straight to those pontine pursuit areas. But from this lateral geniculate nucleus, we have projections back to the striate cortex or area V1, which is Broca's areas 16, 17, and 18, and that's in the occipital lobe. And these here are the optic radiations. And we know that if we have an object in this left visual field, it's going to hit the nasal aspect of the retina in the left eye, and it's going to hit the temporal aspect of the retina on the right eye. These are then going to project back to this right cortex, or these right primary visual areas. So anything in our left visual field is going to be processed more on the right side of our brain. So I think it's important to note here that if we do have an object in this left visual field, it's obviously going to be processed more in this right visual cortex. But if it's also moving to the right, then we have a rightward pursuit. And we know that a rightward pursuit is associated with more of a right cortical stimulation. However, if we had an object in our left visual field that was being processed by the right visual cortex, but it was moving to the left, this would be a leftward pursuit. And we know that that's associated more with these left cortical regions. So basically, if we have something in our left visual field that's moving right, that's probably more of a stronger or more powerful stimulation of this right cortex versus something in our left visual field that is moving smoothly to the left. Okay, so when we've reached this primary visual cortex, we're then going to go to our dorsal and ventral streams. The ventral stream is going to go down, and it is the what stream, and that's associated with the color, the shape, and the size of the object. And we're also going to have the dorsal stream, which goes up, and this is the where stream. And this gives us information on location and spatial relations. For a smooth pursuit, the information goes to areas V2 and V3, and then to the middle temporal and medial superior temporal areas. The middle temporal area is associated with speed and direction of the object, and the medial superior temporal area then extracts this information and gives us information about whether the object is moving or our eyes are moving. We also have projections to the frontal and posterior parietal cortex. So now we've looked at the big picture, we're going to zoom in on the brainstem and some surrounding areas. So we're looking at our patient. You'll have to excuse my poor artistic skills, but this is the right side, this is the left side, these are the eyes, and this is the target moving smoothly to the left. This represents the cortex, this represents the cerebellum. We have the midbrain here, or the mesencephalon, with cranial nerve 3 and 4 nuclei. We have the pons with the dorsolateral pontine nuclei, and the nuclei of cranial nerve 6. And then this is the medulla and the vestibular nuclei. So here we have an object that is moving in our left visual field, and we go from the retina through the visual pathway, we get to the primary visual cortex in the occipital lobe, and then we go through our dorsal and ventral streams, and we end up in the middle temporal area, the medial superior temporal area, and posterior parietal cortex, as well as the frontal eye fields. Again, this middle temporal area is going to be speed and direction of the object, and this medial superior temporal area is going to extract this information, and it tells us whether the object is moving or our eye is moving. So from these areas, we then fire down to the ipsilateral dorsolateral pontine nuclei in the pons. And then from there, we do our first decussation over to the flocculus and paraflocculus, as well as the dorsal vermis. And then from here, the dorsal vermis and the flocculus is going to modulate output of this caudal fastigial nuclei. The caudal fastigial nuclei is going to project to the medial vestibular nuclei, which is one of the 4 vestibular nuclei. And then from this medial vestibular nuclei, we're going to project to cranial nerve 6, and that is our second decussation. From cranial nerve 6 nuclei, we're going to project to lateral rectus on the right via cranial nerve 6, and this is going to fire that right eye into abduction. Cranial nerve 6 also fires to the contralateral cranial nerve 3 nuclei via this medial longitudinal fasciculus. Cranial nerve 3 nuclei then fires medial rectus on the right, and this is going to end in adduction of that eye. So from abducens nuclei, we have cranial nerve 6, which is innervating lateral rectus on the left, and we also have the formation of the medial longitudinal fasciculus, which is projecting to 3 and then innervating medial rectus. So what I've done is I've zoomed in on abducens nuclei to show you that we have these two separate subnuclei, and the alpha motor nuclei form cranial nerve 6, which is going to go and innervate lateral rectus. And we also have here these internuclear nuclei, and this forms the medial longitudinal fasciculus and goes up to 3. The name internuclear ophthalmoplegia, I believe, comes from the fact that we have this group of internuclear nuclei in the abducens nuclei. And internuclear ophthalmoplegia is when the affected eye cannot adduct, and it's caused by dysfunction or injury of this medial longitudinal fasciculus. So next we're going to add in something called the neural integrator. And the neural integrator for horizontal eye movements is the medial vestibular nuclei and the nucleus prepositus hypoglossi. So the neural integrator for all horizontal eye movements consists of the nucleus prepositus hypoglossi and the medial vestibular nuclei. So whether it be a horizontal pursuit or horizontal saccade, if it's holding gaze in the horizontal plane, it all uses this horizontal neural integrator. Again, just to reiterate, all right eye movements are going to use the right neural integrator, and all left eye movements are going to use the left neural integrator. And the neural integrator for vertical eye movements is the interstitial nucleus of Cajal, and that's located in the mesencephalon. So this medial vestibular nuclei projects to the abducens nuclei, but it also projects to the neural integrator on the left, since we're doing a left horizontal pursuit. And what happens is this neural integrator takes a pulse or a speed and it turns it into a step or a position. So the medial vestibular nuclei projects to the left horizontal integrator. This left horizontal integrator takes a pulse and turns it into a step, and then it fires it up to abducens nuclei. So now abducens nuclei has information about pulse or a speed, and it also has information about step or position. So now it has information on where to move the eyes and how fast to move the eyes in relation to the moving target. I just want to add here that much like we have premotor generators for saccadic eye movements, which are the burst neurons in the PPRF, we have premotor generators for a smooth pursuit, and these are located in the medial vestibular nuclei. So we have position vestibular pause neurons located in this medial vestibular nuclei, which project out to the abducens nuclei here. So that's important to remember, that the premotor generators for pursuits are in the medial vestibular nuclei. Next, we're going to add an ipsilateral cerebellar level, and this floccular complex modulates the output of the caudal fastigial nuclei on this side, and this output is going to be inhibitory on the ipsilateral vestibular nuclei. So we're going to have inhibition of this left medial vestibular nuclei, so we don't get a rightward pursuit, so we don't get any excitation of this abducens nuclei, and thus any excitation of a rightward pursuit. For the next layer, we've added a few more structures here in the pons. So we have the nucleus raphe interstitius, which houses omnipause neurons, and we have the pontine paramedian reticular formation, which hosts burst neurons, both excitatory burst neurons and inhibitory burst neurons. So these omnipause neurons are going to inhibit burst neurons in the pontine paramedian reticular formation, and burst neurons are responsible for fast eye movements. And seems we're doing a pursuit, we don't want any fast eye movements, so we need omnipause neurons in this nucleus raphe interstitius to inhibit the burst neurons. So the next question would be, how do we excite these omnipause neurons? And typically we say that there's three things that excite the omnipause neurons, and that's the caudal fastigial nuclei in the cerebellum, as well as the rostral superior colliculi and the frontal eye fields. So now we're going to draw similarities between the VOR and the smooth pursuit pathway. So this here is another schematic of the brainstem, and let's imagine that our patient turned or rotated to the right, stimulating these right horizontal canals. This then fires into the medial vestibular nuclei, across the abducens nuclei, and then down via the MLF to 3 here, and then medial rectus is innervated by cranial nerve 3, and lateral rectus is innervated by cranial nerve 6. So everything in this green arrow is part of the VOR. So the only thing that isn't in the VOR is this extra bulbar or this cortical portion. So this VOR fires into the medial vestibular nuclei and position vestibular pons neurons discharge from here and fire cranial nerve 6, and we go through that same pathway, and this latency is about 5 milliseconds. So if we see a person that has issues with a leftward pursuit, then we'd come and we'd test the VOR, and if the VOR is intact, then we know that most everything in this circle is functioning pretty well, and it must be something outside of this VOR pathway that is causing these breakdowns in pursuit. So the final layer here is to add in this structure, the NRTP, which stands for the nucleus reticularis tegmente ponti. It's also in the pons. It doesn't generate eye movements, but it integrates into the brainstem generators. It's a major integrator into these pontine oculomotor areas, as well as the dorsal vermis. So we have firing down from the frontal eye fields and the supplementary eye fields to this ipsilateral NRTP, and then that's going to fire bilaterally to the dorsal vermis or the oculomotor vermal area. And this dorsal vermis is going to modulate its output again on the caudal fastigial nuclei. On this right side, we're going to have excitation of this medial vestibular nuclei, whereas on this left side, we're probably going to have inhibition of this medial vestibular nuclei, so we don't generate that rightward pursuit.
So this is what that left pursuit looks like overall, and it appears messy and complicated, and it is, but we have broken it down layer by layer and gone through each one in detail. So we have areas involved in calculating speed, initiating and stopping the pursuit, maintaining motion, ignoring a background, and prediction mechanisms or estimation mechanisms. And the moral of the story is that there's a lot going on in this pursuit. And when it comes to vertical pursuits, the only real difference is that the neural integrator for vertical eye movements is the interstitial nucleus of Cajal, and that's located in the mesencephalon. And the burst neurons that must be inhibited are located in the rostral interstitial medial longitudinal fasciculus. So as I mentioned, this pathway is complex, and anatomically it's long. So that means it's susceptible to head trauma. Head trauma like mild traumatic brain injuries, for example, a concussion, they sometimes involve tractioning of axons. And Dr. Carrick uses the analogy, it's like wringing out a wet towel. And when you wring out a wet towel, the greatest force is right in the middle. So the more midline primitive structures get affected, just like the ones we've been talking about in these pursuit pathways. So to finish off, we're going to go through a few patient scenarios. So let's say that our patient presents and we test pursuits on them and they have poor leftward pursuit. These are some of the things we want to be thinking about when it comes to our differential diagnosis, and then we're going to compare that to our history and our exam findings. So firstly, we need to ask, can the patient see? How is their vision and visual acuity? Then next, we're going to look at the cortical areas. For example, if we have sensory findings from our exam, we might be more suspicious of something going on with the parietal eye fields. Next, we can look at the cerebellum. How are there other cerebellar findings like rapid alternating movements? And then from there, we can go deeper into the cerebellum and differentiate dorsal vermal lesion versus a caudal fastigial nuclei lesion. And we can do that by looking at a patient's saccades. A dorsal vermal lesion will typically give you ipsilateral hypometric saccades, and a caudal fastigial nuclei is going to give you ipsilateral hypermetric saccades. So then after that, we can come and check the vestibular nuclei. Are there any cranial nerve 7 or cranial nerve 8 findings? And then how is their VOR? We know that a pursuit is essentially a volitional VOR, so we could potentially do a yes to rehab a vertical pursuit, or a no in the horizontal plane for a horizontal pursuit, because these pathways are so similar. Then we're going to come and look at cranial nerve 6 and cranial nerve 3 and see if there's any correlation between other eye movements, for example, convergence. And then finally, we're going to look at the neural integrator. Is it just the pursuit that is bad, or is it all horizontal eye movements? If so, we could have a neural integrator problem. So then we can check to see if a certain eye movement improves another eye movement. For example, does a leftward saccade improve a left pursuit? If so, maybe the leftward saccade is priming the neural integrator and we can then use it to rehab the pursuit. So that's a few scenarios here. Then next we're going to talk about saccadic intrusions. So if we have a patient who has saccadic intrusions, we have to ask, why are burst neurons in the reticular formation firing and why aren't the omnipores neurons inhibiting them? What happens when we activate the caudal fastigial nuclei, the superior colliculi, or the frontal eye fields, which are the areas that are supposed to activate omnipores neurons? Burst neurons are in the reticular formation and they're closely associated with the autonomic nervous system. So whenever we have inappropriate firing of burst neurons, we always have to check autonomics. The next situation is going to be catch-up saccades versus square wave jerks and how we differentiate between the two. So catch-up saccades and backup saccades as well can be a compensation mechanism for an increase or decrease gain with pursuits. So then we have to ask, why are we having increased or decreased gain with the pursuit? And we could potentially use a ×0 or a ×2 VOR to rehab that. Catch-up saccades are different to square wave jerks. So square wave jerks, especially in the direction of the pursuit, is an inappropriate response rather than a compensation. It means that burst neurons are firing when they shouldn't be. So again, we have to look at trying to encourage the inhibition of burst neurons or figure out why these burst neurons aren't being inhibited. The next situation is going to be a patient that has nystagmus, and nystagmus is essentially burst neurons firing and omnipores neurons not working like they should. If you have gaze-evoked nystagmus and also nystagmus during pursuit, then we can theorize that it's more of a gaze holding problem rather than a pursuit problem. So you probably have to rehab gaze holding before fixing the pursuit. If you have poor pursuits but gaze fixation is fine, it's probably more of a pursuit mechanism. So there are a few patient scenarios or clinical scenarios where we can start thinking about this stuff from a more clinical perspective. If you have a good understanding of these pathways, then hopefully it's going to help you next time you're looking at a patient or looking at a patient's horizontal pursuits. So we can be more accurate in our diagnosis of the functional deficit or the lesion, and thus more specific in our treatment to ultimately better help our patients. As we've seen in this video, the pursuit pathway is complex and intricate. It involves an interaction of many different regions of the brain, and these regions play a crucial role in eye movements, but not only that, they play a crucial role in other human functions. So observing and measuring these eye movements can give us a great window into the functionality of the brain, and by improving the function of brain regions to produce a smooth pursuit, we can almost guarantee we have made a positive change to other human functions. So, thank you for watching. I hope you've enjoyed this video and learned something, and I will see you next time for our video on saccades.